So again, we are really delighted to have Dr. McClure with us today. Currently, Dr. McClure is professor and chair in the Department of Epidemiology and Biostatistics at Drexel, as well as adjunct professor of biostatistics at University of Alabama in Birmingham. Prior to coming to Philadelphia in 2015, she was professor of biostatistics at University of Alabama in Birmingham. She obtained her PhD in biostatistics from University of Michigan in Ann Arbor and an MS in preventive medicine and environmental health from University of Iowa. She has a very long list of publications. I counted them, more than 150. And a very large number from the regards cohort, and it's very close to my heart because I have a very good friend who worked on this regards cohort. It's the reasons for geographic and racial difference in strokes at a cohort that started at University of Alabama, I don't know, 15 years ago, approximately. Yeah. And they did an incredible work, epidemiological work, absolutely amazing with these patients, both for chronic kidney disease and stroke, obviously. She is also the PI or co-investigator on multiple NIH grants and is a teacher and mentor to numerous master and PhD students, and has been a reviewer also in multiple NHA, NASA statistics sections, and lots of work. She also did lots of work on environmental issues. So thank you again, and we are absolutely excited about learning more about clinical trials.
Thank you. Thank you for that introduction and for the invitation. And I have to warn the people who are watching that I'm a wanderer. So if I get from behind the podium, I apologize. I'll wander back at some point. Also, I'm not going to talk about the ReGARD study today. I'm going to talk about some clinical trials work I've done. But maybe in the future I can come back and talk about that. It's an amazing study, an amazing cohort, and we are always looking for collaborators. So I'll say that. But thank you again for the invitation. Today I'm going to talk about implementing a mid-trial adaptation in a stroke clinical trial, the good, the bad, and the ugly. So first of all, I have no disclosures. Let's see if I can use this to advance the slides. No. So here are some learning objectives for the talk today. So by the end of the talk, I hope you'll be able to understand the rationale behind sample size re-estimation, describe the impact of sample size re-estimation on SPS3, and describe the different aspects of the study that were impacted by sample size re-estimation. So I'm going to tell you a story today about a clinical trial that I was involved with. I'm going to start with some background and then introduce the trial itself, and then talk about this mid-study adaptation we did, and then the impact of that. So I'll kind of hit lots of different aspects of the study, including the science behind it, the statistics behind it, and then some of the more practical trial management issues. So according to the CDC, in 2014, the last year that there were published data, which might be the last year we ever have published data, stroke was the 5th leading cause of death in the United States. And it's the #1 cause of long-term disability among adults in the U.S. as well. About a third of stroke survivors suffer from another stroke within 5 years and 14% within 1 year. So the impact of stroke is considerable in the U.S. Understanding how to prevent stroke is a topic of much research, but stroke is a somewhat rare disease. So in order to look for risk factors or impact risk factors for first stroke, you need a large study like ReGARD, which has 30,000 people. So a lot of attention has been given in clinical work to how to prevent recurrent stroke among those who've already had a stroke. And as many of you probably know, antihypertensive medications are recommended for secondary stroke prevention among those who've already had a stroke or a TIA. And lifestyle modifications are recommended as well, such as quitting smoking, improving your diet, and getting exercise. Additionally, antiplatelet therapy is recommended for prevention of recurrent strokes for those suffering from specific stroke subtypes. So the literature is really mixed on this depending on the type of stroke that the patient has, but different treatments such as aspirin or Plavix or different antiplatelet therapies and combinations are often used, particularly in the acute setting, to prevent recurrent strokes. However, at the time that this study I'm going to describe was implemented, there was a lot of question about how far blood pressure should be lowered to best prevent recurrent stroke. So people who have stroke are often given antihypertensive medications, and the prevailing thought was the lower the better. But there wasn't really any evidence to really inform that idea. And then further, in certain, as I mentioned, in certain stroke subtypes, there was some discussion about which antiplatelet therapy or combination therapies were most effective for reducing the risk of recurrent stroke. So the secondary prevention of small subcortical strokes was designed to help address these issues. So SPS3 was designed to assess stroke recurrence among patients who had already had a lacunar or subcortical stroke. These are mild strokes. Patients who have a lacunar stroke often have fewer complications than patients who suffer from other types of strokes. And you can see in the picture where in the brain a lacunar infarct occurs. Now, I'm a statistician, so I've just given you my entire scope of what I know about stroke. Okay, so SPS3, the Secondary Prevention of Small Subcortical Stroke Study, was a multinational, multi-center randomized clinical trial with goals of assessing two things in particular: whether the combination of aspirin and clopidogrel or Plavix was more effective at reducing the risk of recurrent stroke than aspirin alone. So an antiplatelet intervention that was looking at combination therapy versus aspirin alone. And then whether the risk of recurrent stroke would differ for those randomized to two blood pressure targets, what we called at the time the usual target range, which was 130 to 149 millimeters of mercury, or what we called the intensive target range, which was blood pressure lower than 130. So I should add that this study was completed prior to the completion of the SPRINT trial, which also looked at blood pressure targets among people with cardiovascular risk factors but not diabetes, and used similar blood pressure targets and actually a similar blood pressure control algorithm to what we used in SPS3. Okay, so SPS3, oh yeah.
Quick question.
Yes, please.
What time period was used to lower the blood pressure?
So the algorithm was that we started to treat, and then they came back every 2 weeks until blood pressure was in target, and then we saw them every 3 months to adjust their medications. And if I remember correctly, the first step was to use diuretics, and then they added on from there. But it was left to the discretion of the hypertension specialists at each site. So the study had their suggested algorithm, but each site was free to treat the patients how they saw was best. Okay, answer your question. I think it's similar to what was done in SPRINT, if you're familiar with SPRINT. So we used a 2x2 factorial design, and we randomized patients to one of 4 groups: one of the 2 blood pressure control arms and one of the antiplatelet therapy arms. So each of the patients ended up in one of these 4 cells. SPS3 worked because of several different parts that were necessary. So the most important were the clinical sites. We had 82 clinical sites that participated in the study, and this is where the patients were recruited and followed. They were responsible for entering their data and transmitting it to the statistical center, and each site was responsible for doing an MRI. Each stroke was MRI confirmed so that we could ensure that it was a small stroke. The Clinical Coordinating Center was located originally at University of Texas Health Sciences in San Antonio, but ended up in University of British Columbia under the direction of Oscar Benavente. And the Clinical Coordinating Center managed the day-to-day operations of the study and the budget, convened the steering committee, and oversaw the general protocol, and also read the MRIs centrally. The Statistical Center was located at UAB under my direction, and we were responsible for ensuring data quality, for designing and developing the data entry system, performing the randomization algorithm, reporting both to the study committees and back to the NIH, and also the analyses and reporting of the trial. And then we used the Drug Distribution Center from the VA Cooperative Studies Center in New Mexico, and they were responsible for purchasing and maintaining inventory for the drugs and also supplying the drugs to the clinical sites. So a lot of different moving parts working together. in a lot of fun. Okay, so I'm going to talk a little bit about how we started off in SPS3. So the sample size originally was determined to be 2,500 patients, and this was due to the following assumptions. So we planned to do our outcome to be the time to recurrent stroke, and this was defined as either an ischemic or a hemorrhagic stroke. And so our planned analysis was a Kaplan-Meier or log rank test to account for our survival data. We assumed originally, based on data in studies prior to the beginning of SPS3, that we'd be following patients for 3 years on average, and that we would have an annual recurrent event rate of about 7% in the aspirin group, and that we would have about a 10% loss to follow-up, which would be about 3% per year. And we allowed for 2 interim analyses during the course of the study. The study design was based on data in the literature at the time. So these rates, this annual event rate of about 7%, was based on mostly on a different study called the WARS study. So given those assumptions and that sample size, we would have had 90% power to detect a 25% relative risk reduction attributable to combination therapy, which would mean that given the 7% event rate in the aspirin group, that would mean a 5.25 annual event rate in the combination therapy group. And similarly, we would have 80% power to detect a 20% relative risk reduction attributable to combination therapy. So those were our assumptions, and this is how we came to 2,500 patients. SPS3 was designed in the turn of this century, around 2000, and the pilot study was going on about 2001. And we randomized our first patient in 2003. We did similar calculations for the blood pressure arm. There really wasn't a lot of information in the literature about what we might expect in terms of recurrent stroke rates for those 2 targets. So we made similar assumptions. And we did not assume that there'd be an interaction between the blood pressure arm and the antiplatelet arm. So we didn't assume that the effect of the antiplatelet therapy would differ for those in the higher target compared to those in the lower target. So we didn't power our study to detect a significant interaction. And this is important because from a statistical point of view, you need about 4 times as many patients if you assume there's an interaction. And so, we had 2,500, and so we'd be talking about 10,000 patients. Okay, so as I said, we started SPS3 in 2003, and we based our calculations for our sample size on the WARS study, which was published in 2001. Since the time that SPS3 was planned, though, several studies started coming out that were publishing stroke recurrent rates that were lower than what we planned in SPS3. So you can see these 4 studies here, and the y-axis represents the stroke recurrence rate in their different arms. And so none of these studies actually had the same interventions as SPS3, but several of them did look at aspirin alone, and some of them were in different populations. But you can see that overall, these stroke recurrence rates seem to be much lower than what we planned. Remember that we planned a 7% recurrence rate among our monotherapy patients and a 5.25% reduction in our recurrence rate among our dual antiplatelet therapy patients. So we're looking at perhaps that the rate of recurrent stroke is going down. And actually, somewhere along this timeline at the stroke conference, someone actually presented a paper showing that in clinical trials, recurrent stroke rates were declining. So this caused some concern among our investigators and our team, because smaller than expected event rates could result in an underpowered study. And so this was a substantial investment of time and money on both the study staff, the patients, and also the NIH. And we wanted to make sure that we had the best chance of seeing a significant result if one actually did exist. So our steering committee petitioned the Data Safety Monitoring Board, the DSMB, requesting that the study investigators be allowed to know the overall event rate. So each NIH institute operates differently, and in some institutes, the way their clinical trials are run, the investigators have a lot of access to data and get a lot of information about what's going on in the study. The Stroke Institute, NINDS, is actually has an unwritten rule that investigators are not allowed to have much involvement in the data at all and are not allowed to know the overall event rate in the study. Their thinking is, well, from my perspective, they're worried that there could be even the perception of bias. So that if they know that the overall event rate is lower, perhaps, than they expected, then maybe they're going to start recruiting patients that don't meet the entry criteria quite right, just to maybe get that event rate up. So at the point in the study when these papers started to be published, I should say, the clinical investigators were unaware of what the rate of stroke recurrence was in the study. So we petitioned to our data safety monitoring board to ask if we could know that, and the DSMB accepted the request. Yes?
So how many years in the study were you at that point?
I think we were about 2009. So remember, we were going to have an average 3 years of follow-up. And actually, in the end, we have about an average of 3 years of follow-up, but recruitment took incredibly longer than we had anticipated. And so we were at about 2009, I believe, when we implemented all of this that I'm going to describe. Yeah, it's a good question. I mean, we were very naive, and I can say that easily because I actually came to UAB right as the study was starting, so I can say we, meaning they were naive when we designed the study to think that we would recruit as quickly as possible. But it was based on our pilot data, so we just didn't anticipate some of the challenges we had implementing sites all across the country and also in other countries. So at this time, the DSMB let us see the overall event rate, and it was 3.4% per year. Okay, so much lower than what we had anticipated. And so the DSMB recommended to us, after some discussion, that we use this information to assess the impact of modifying the study design in order to complete the study with sufficient power. Okay, so one of the new buzzwords in clinical trials is adaptive designs. And so adaptive designs are the process of making adaptations, making changes to your study mid-course. And there are a lot of different ways to adapt your study. Some of the more popular include like adaptive dose finding for earlier phase trials, or other types of dose or treatment adaptations where you start with a lot of treatments and then you start reducing them down depending on the evidence. And you use the evidence in your data up to that point in the trial to make these decisions. And so from the perspective of you don't really ever know what's going to happen in your trial, there's a huge benefit to it because as you're going along, you can make changes. From a statistical perspective, there could be issues. And so we wanted to determine whether increasing our sample size or extending our follow-up time would impact the power of our study as well as the type 1 error. So our goal was to see if we had more patients or if we followed the patients longer, would this give us enough power? Would we see enough events to be able to detect that difference that we had anticipated? But the worry is the more you look at your data, the more likely you are to say there's a difference when there isn't really a difference. And so we wanted to be able to ensure that making these changes would not impact the type 1 error rate. And so we used statistical simulation to examine this. And so we did the following. We looked at 4 different adaptations. The first was just our planned study design. So originally we planned 2,500 patients with 1 year of follow-up. After 1 year of additional follow-up after So we looked at the planned study design with an additional year of follow-up from the time the last patient was recruited. The second was to increase the follow-up time. So still 2,500 patients, but now we're going to look for 2 years after the last patient is recruited. The second 2 adaptations were to increase the sample size and then to 3,000 and then follow for 1 year after the last patient was recruited, or increase the sample size to 3,000 and follow for 2 years after the last patient was recruited. So we sort of have this 2x2 factorial design for our simulation. Yeah.
Is your assumption that the attrition rate is the same over the entire year?
The attrition rate?
Yeah.
Yes. So I'm going to talk about on the next slide, actually, or maybe it's on the next slide. But we assumed all of the things we were seeing in the study at that time are going to continue. So we assumed or I'll talk about it on the next slide, but the recruitment rate we assumed would be the same as what we saw, or maybe a little slower, or maybe a little faster. We assumed that the event rate would be what we are currently seeing in the study. So remember, the overall event rate was 3.4% per year at that time. So initially, we assumed that it was 7% in the aspirin group and 5.25% in the combination therapy group. And that's a 6.25% overall rate. So if we average these two. So if you look, we were almost at half the event rate that we had anticipated. So we assumed the planned reduction in recurrent stroke. So we assumed that this difference, this 25% reduction, would hold. And so that gave us a 3.9% event rate in the antiplatelet, or in the aspirin and placebo group, and a 2.9% reduction or rate in the combination therapy group. So we kept this 25% recurrent relative risk reduction in the recurrent stroke rate to get back to this overall rate of 3.4% per year. Does that make sense? Okay. Okay. And then we assume patients entered at the study rate similar to those observed in SPS3 at the time. So at that time, we were recruiting about 25 patients per month. And so we looked at 25 patients per month, but we also looked at 20 per month and 30 per month. So the faster you recruit, the quicker you get to the end of the study. And so there's implications for the speed at which you recruit as well. And so one of the things that happened in SPS3 was we recruited at a much slower rate than we had thought, which meant we were following patients longer than we thought. In the end, we had one patient with like 9 years of follow-up. Okay. Yeah, so we had assumed an average of 3, right? So this table presents the type 1 error rate. So this would be the likelihood of seeing a result when we, in fact, don't have one. You'll notice on this table, the far left column is sample size, and then the number of patients per month, and the number of additional years of follow-up. And I only have 3 of the cases here. After looking at the situation where we recruited 3,000 patients and followed them for 2 additional years, it was just beyond the scope of what was feasible for the timeline for the study. And so what we found was that regardless of the adaptation we made, the type 1 error rate, which should be about 5%, was maintained. And we only had one case that it was a little bit higher, but that was still within the confidence interval around 5%. So we felt confident that we could make this adaptation, make any of these choices, and we wouldn't impact our type 1 error rate for the study. This figure shows the average power. This is assuming the overall event rate. And so what I'm presenting here on the y-axis is the average power. On the x-axis is the number of patients we recruit per month. And each column represents a different number of patients recruited. So the red bar is 2,500 and following them for 1 additional year. The yellow bar is 2,500 and following them for 2 additional years. And the red bar is 3,000 and following them for 1 additional year. And what we saw from this was what we would expect, first of all, that the more quickly we recruited patients, the lower the power. And that's because our timeline shrinks. If you recruit your patients faster, you finish your study more quickly, and there's less time for patients to have an event, frankly. But not considerably so. What we also saw was that we gained more by recruiting a larger number of patients and following them for a shorter time as compared to recruiting 2,500 and following them for 2 years. And we also felt happy to see that our power increased. So we went from about 70% to roughly 80% by doing this adaptation. Okay, so the 2 things we were concerned about, type 1 error, we were protected, and power, we should have sufficient power, we felt relieved that this could work. So after we engaged in this simulation study, we looked at several different cases, and we discussed the results with our steering committee. The steering committee recommended that we recruit an additional 500 patients and then follow them for an additional year after the last patient's randomized. So 3,000 patients. And our conclusion was that it didn't impact the type 1 error rate and that it improved power from the current design to about 75% with a range of 66 to 82%. And the way we got that range was we actually looked at the confidence interval around the event rate in the study at the time, and we repeated the simulation study for the lower and upper bound of it. So that's what we figured was about our bounds for our power. And so the study modification was approved by the NIH, and it was implemented after really, really lengthy discussions among the investigators. And so I'm going to kind of switch gears now a little bit to talk about the implementation and then the results. Are there questions about the sample size re-estimation before I do? Yes.
I guess I'm not sure I understand. The 75% is your new power. It's
Yes.
your expected power.
That's our expected power given our new sample size, given the overall event rate at the time, to detect that 25% relative risk reduction.
But don't you want to get to the 80%?
We couldn't. So we could, but it wasn't feasible in terms of the design parameters. So to get to that 80%, we would have had to keep going for much longer than the NIH would be willing to do. So, but there is no magic about 80% power. It's just it is a commonly accepted threshold. But the reality is that 75% is not bad. And really, if you look back at most clinical trials, even though I highly disagree with doing what's called post-hoc power calculations, most of them are way, way underpowered to detect the difference that they originally anticipated. So we were satisfied with 75%. I think had we been 60 or below, I think we would have been concerned. But with 75%, I think we were satisfied. But it's a good point. I mean, we're not quite at 80. We just couldn't get there within the scope of what was reasonable, given how long we'd been going already. So, other questions? Okay. So, in addition to the statistical concerns, there were numerous practical issues that needed to be addressed. In fact, the simulations were fun to do and took time, but the implementation was extremely difficult. So remember, we originally planned the study to be about an average of 3 years of follow-up. And so most of the IRBs at the sites planned for a 5-year study. And so many of the sites we had to change the protocol, which led to many of the sites having to update their IRBs and their contracts. We had to extend patient follow-up, and so our original informed consent only provided consent for a 5-year study. So for some patients, we had to reconsent them. And this did impact attrition. We did have some patients who said, enough is enough. I don't want to do this anymore. Now, the flip side of it was that they were all getting their blood pressure medications for free. So many of them were like, yeah, sign me up. I'll keep going as long as you want me to. And we saw the patients every 3 months, and so the study staff had great relationships with the patients, and they were used to getting regular care. So that was a little less difficult than it could have been. We had to change our forms. So we have 40 study forms, and at the top of each one, there's the study visit, and it only went up to the 5-year visit. So we had to change our forms, which then meant we had to change our data entry system, which then meant we had to change all our statistical programming code to account for this extended variable. We had to get funding. So we were only funded for a 5-year study, and we had to ask the government to continue to fund us, and luckily they did. And I'll talk a lot more about the funding in a little bit. We needed to extend contracts for our study personnel. So all of the study staff are on contracts. Many had started looking for jobs already as they thought the study was coming to an end. And so we had to retain and update contracts. And then we really, really had to address issues of study fatigue. So there were patients who had been in the study at the time 5 years and really had to figure out ways to continue to engage them besides just getting free drugs. But not only for the patients. We had to address issues of study fatigue for all of the study staff as well. Some of the coordinators, many of the coordinators had worked on a number of different studies, and they liked the idea that they were, doing different things over time, and to encourage them to continue to help recruit. And the PIs, the site PIs, needed They also would like to move on to different studies and be involved, and they needed to recruit patients. And even the staff in my group, we really, people have been doing the same thing for a long time, running the same reports every 3 months. And so really addressing issues of study fatigue were really important. Okay, so then what's the rest of the story? So we closed recruitment in April of 2011 with 20 patients enrolled. So we hit our target and then some a little bit. And here's where they all were. So you can see the majority were in the United States, but we also had a lot of Latin American and Spanish patients who were enrolled in the study, and Canadian as well. So I didn't talk about, there were many issues not only with extending the study in the US, but internationally as well. And everything we did, we had to translate to Spanish and French. So anytime we had to change something, it had to be translated. But we were very excited. It was an exciting day when we completed randomization. Okay, and then what happened? So we completed randomization in April, and throughout the course of the study, we at the Statistical Center had noticed on several occasions that there was no efficacy for the combination therapy antiplatelet arm. And in fact, so we expected that there would be higher rates of bleeds among those on combination antiplatelet therapy. So mostly GI bleeds, but other bleeds as well. And so we'd been seeing that, and along the course of the study, that threshold, that difference never crossed the threshold that the Data Safety Monitoring Board had deemed to be unsafe. So we'd always been within that range that we expected based on what was known about combination therapy. But we started to see the risk of death creep up a little bit among those who were on the antiplatelet therapy, and no efficacy. And so we at the Statistical Center were very confident every time we had a DSMB meeting that they were going to shut down that arm. And they never did. So then we come to July 2011, and we had a DSMB meeting, and we were confident that they were not going to make any changes to the study because we were so close to completing the study. However, they thought differently. And so the decision was made to terminate the antiplatelet intervention due to concerns with safety and a complete lack of efficacy. So we closed out that arm. So we just made all these changes, just finally completed randomization, and now we had to make all these changes, and we had to get all the patients in and close out that arm, but continue them on the blood pressure arm. And so the results of the antiplatelet trial were published in the New England Journal of Medicine, and then follow-up continued on the blood pressure arm until April of 2012, and the results were subsequently published in The Lancet. So because of the way the study was designed, because of the rolling enrollment, we decided it would be interesting to take what results we saw with the 20 patients and compare them to what would have happened had we stopped with 2,500. So just a quick, I'm going to show the results in a second. But what we found was that there was no efficacy for the antiplatelet arm. So there was no difference in the recurrent stroke rate between the combination therapy and the monotherapy. And what we found in the blood pressure arm was that blood pressure lowering was associated with a non-statistically significant lower rate of recurrent stroke. And that p-value was 0.08. So it was close, but not quite there. And so we were curious, what would have happened if we would have stopped at 2,500? Because we had all of these administrative challenges, as well as funding that allowed us to continue. So we were curious, what would have happened if we would have stopped with those initial 2,500? So we compared the results of what we saw with the 30-20 to what might have been had we stopped at 2,500. And we looked at both the safety and the efficacy. And so remember that our primary efficacy endpoint was this composite of any recurrent stroke, either ischemic or hemorrhagic. And our primary safety endpoints were major bleeds and death. And we also considered the costs of extending the study. Okay, so in this table, I'm presenting what we saw at the end of the study. This is the comparison of the primary results for the antiplatelet arm, and this yellow block is what we saw at the end of the study. These were our published results. This is the result had we stopped at 2,500 patients and stopped their follow-up 1 year after the last patient was randomized. So these are published now because this whole thing has been published. But these were our published results in the New England Journal. And so what we saw was that the number of events, of course, increased longer follow-up, more patients. The event rates really, or the average follow-up really didn't change. And this was because of that closure of that arm early. And then you can see the event rates are pretty much the same, and the hazard ratio was 0.92, not statistically significant, and 1.0 had we stopped earlier. So, and the p-value, had we stopped earlier, would only have been higher. Okay, so we didn't see any difference in the results due to our sample size re-estimation. And we're not surprised. I mean, as I said, we knew all along there was no efficacy in this arm, or there was no suggestion of efficacy. Okay, so this figure, or this table is the same thing. Now this is for the blood pressure intervention. Okay, so again, this is what we published in the Lancet. And you can see here, this was our hazard ratio and our confidence interval p-value, so our 0.09, 0.089. And this is what we saw, what we would have seen had we stopped with 2,500 patients. So again, of course, fewer events, shorter follow-up time, slightly different event rates, but not dramatically so. The hazard ratio was a little higher, but not incredibly so, but was less significant. So we definitely did gain power for this blood pressure effect by extending the study. However, it wasn't quite enough. We didn't quite make it. 0.089. Okay, so I'll talk about what that meant in a minute. These are the safety results, and what this is set up a little differently. The yellow panel is still the 30-20 patients at the end of the study, what was published in our primary publications. The blue panel is still the 2,500 patients, but now I'm just presenting in the top two rows the antiplatelet intervention, and in the bottom two rows the blood pressure intervention. And these are just the hazard ratios and 95% confidence intervals. And what we saw was that for major bleeds and death, both for the antiplatelet arm, there was no difference. And for major bleeds and death, for the blood pressure arm, there was really no difference either. And you can see this increased risk of both major bleeds and death in the antiplatelet arm, where there was higher risk of both death and major bleeds among those in the dual antiplatelet therapy. And again, we expected this. We expected that this would happen. It wasn't as dramatic to see that. This was a little bit more of a surprise. And that was what led to the early stopping for that arm. Okay, and this is just that information graphically. And so if you look here now, these are the primary events, and the left is the antiplatelet intervention, and the right is the blood pressure. So you can see these orangish dots are the 30-20, and these greenish dots are the 2,500. So you can see how they shifted a little bit, but no results really. The clinical interpretations really didn't change much at all, or so I thought. Okay, so I'll come back to what this meant for the results in a minute, but let me talk about cost. So each site received $4,000 for each patient they randomized and completed a baseline visit, and for each follow-up, they received a $400 reimbursement as well. So with this extension, we had 520 additional baseline visits, roughly, and 2,583 additional follow-ups. Remember, we were following them every 3 months. And so that resulted in an increase of $3 million just in patient costs. Once you factor in all of the study costs, including the medications, the statistical and coordinating center costs, the drug distribution center, we estimated that it was about $21,000 per patient to run this trial. So this resulted in about $11 million increase in costs to do the sample size re-estimation. Okay, so the good. What was good? Well, the antiplatelet results didn't change appreciably with the addition of the 520 patients. We still saw a null result for the primary outcome. We are still detecting these safety concerns, and so we weren't masking anything. There was nothing that we changed in our interpretation. These results are highly confounded, though, by the early closure of this arm. So given the actual treatment observed, I'm pretty confident that we wouldn't have seen anything if we had gone on forever with this study in the antiplatelet arm. But there is that caveat that we stopped it early, so you don't always know. the blood pressure efficacy results trended towards significant at the end of the study, and it was a really interesting discussion among the investigators. So had we stopped the planned study end, we would have had that 0.4 p-value, and we would have had a clearly negative trial. There would have been no question that there was no difference in the recurrent stroke rate, regardless of the blood pressure arm. However, we saw a 0.09, 0.08 p-value, and there was lots of discussion about whether or not this was significant or clinically relevant. So the discussion, the statisticians, we are kind of more conservative, I guess, in some ways, and we talked about things like we did multiple interim analyses that weren't accounted for. We had these two arms of the study. I studied multiple comparisons and multiple testing for my dissertation, so I'm a real stickler that we didn't actually account for all of the multiple testing we did, and so this 0.089 should actually be a little bit higher. But the clinicians among us felt very comfortable saying that there was a trend, and it was definitely suggesting that the blood pressure, the lower blood pressure arm was better than the higher.
Yeah, but the confidence interval was pretty wide. I mean,
it was to 1, yeah. So definitely the trend was there. However, the strength of that association is hard to know. And we didn't achieve the clinically relevant difference that we had planned. So we had anticipated a 25% reduction in recurrent stroke, and we didn't achieve that. So it's also hard to know what is clinically relevant. How much do we need to see to be able to say that, yes, this is important? And so actually that wasn't something that was brought up by the reviewers when we published the primary paper, but when we published this, the reviewers in the Clinical Trials Journal said, but you didn't hit your original target. Is this still an interesting clinical difference? And so it did lead to interesting discussions. And now actually the SPRINT trial was actually stopped early because there was such strong efficacy showing that lower blood pressure target led to fewer events. And their primary outcome was major vascular events. They were looking at cardiovascular or heart attack, stroke, and death, I believe. So there is now really concrete evidence that lower blood pressure is better for certain populations. So it could also be that we were looking at lacunar strokes. They're fairly mild strokes, and perhaps there wasn't the same impact on this population as there might be on different populations. So the safety results for the blood pressure arm were not impacted, and we also did look at some hypotension and other events you might expect when lowering blood pressure, and we really didn't see any difference, first of all, in the two arms, but also any impact from the study modification. So the bad, in my perspective, is that there was a lot we had to do to implement this, and it's often overlooked. So the practical changes to study management, the changes to the forms and the databases, personnel changes. Most of the literature about sample size re-estimation and study adaptations really focuses on the statistics. And will we be able to still detect differences that we had planned? But very little talks about the impact on the practical aspects of the study. This was extremely taxing for the study staff all around. And I mean, of course, there's no way to know that you're not going to have a significant result in the end without doing it, but it does beg the question of, did we do all of this and was it worth it? And then, of course, the ugly as a taxpayer is that the huge amount of increase in costs compared to the small gains in statistical power. And so was this cost effective, and is there any way to know when it will be cost effective to do the adaptation? And those are questions we really can't answer. I mean, again, most of the literature focuses on the statistics, and very little talks about the cost of implementing these changes. And so you might think about like other types of adaptations, and maybe it's not a big deal. But for instance, what if you're eliminating arms? So every time you eliminate an arm, you have to change the randomization scheme. You have to make sure that the drugs that they're being randomized to, those that got eliminated are no longer available there to be grabbed by accident. You have to make sure that your programming is updated to reflect these changes. So there are still downstream costs and implications that make it difficult to understand if these are cost effective. And I know when I present this to clinical trials audiences that are largely statisticians, I'm always afraid I'm going to get rotten tomatoes thrown at me because I feel like I'm standing up and saying, yes, we're all developing these new statistical methods for adaptive designs, but why? I mean, is there really good reason to do this? And so some discussion. So this is definitely a popular trend in clinical trials, and I'm on the bandwagon. You know, I have 2 students who have looked at other approaches for sample size re-estimation and other adaptations, but it's really, really important and it's important to do this because we very rarely get our assumptions for our study correct. We very rarely know. If we knew, we wouldn't have to do the study. So it's really important to be able to look midstream to see the impact of your assumptions on what's actually going on. And in SPS3, I do think that, well, maybe the sample size re-estimation helped to provide a muddier answer as to which level of blood pressure control was more effective in preventing the recurrent stroke. But had we stopped at the first point in time, we wouldn't have even been having the discussion of whether there was some clinically relevant difference. But it's really not clear, again, that gain in information is offset by the increase in costs and time. And this is just one example of a use of sample size re-estimation where the study modification did not materially change the outcome, but there are others out there where there have been positive impacts of doing the sample size re-estimation. But I say again that as the use of adaptive designs increases, and I'm reviewing grants every cycle now, where there's, I'd say 50% of them for these large phase 3 studies, and often for maybe 25% for the phase 2, have these adaptive approaches incorporated. We have to pay more attention to the statistical characteristics, but also how the operational characteristics will change and the costs. So it's this balance, and we're not really sure how to do that a priori yet. And so just want to end by acknowledging my collaborators. Doctors Chris Coffey and Jeff Zykowski were our biostatisticians who I worked with at, Chris is at Iowa, used to be at UAB, and Jeff is at UAB. Of course, Dr. Benavente, who's the PI of the Clinical Center, and Bob Hart, who was the co-PI of the Clinical Center. The steering committee and the DSMB, we had many, many interesting conversations about this, and it was very fascinating scientifically. And then, of course, the study participants and the NINDS for funding us for many years. So thank you, and I'm happy to take any questions if you have them. Yes?
So there's a number of issues I'd like to mention, but just one. I really thank you for your presentation. That really enforced my negative view towards adaptive design. So one of the interesting things about the 0.08 is that, to me, that sort of demonstrates kind of a lack of understanding on the part of clinicians about what a p-value really means. And if you have, in my view, if you have a randomized trial, then if you set your p-value at 0.05, then it either is or it isn't. There's no such thing as a trend.
I agree, and to me that represents a lack of poor teaching on my part. We are not doing a good job educating clinicians about what that is, how to interpret.
Yeah, I mean, I know it's very difficult to do it, and it's so pervasive that assume that a p-value imports importance to the... But
it's also difficult after 12 years, you know, to stop and go, oh my God, we didn't get anything to show for it. So, but yes, I think, so that tells me we're doing a bad job as statisticians teaching clinicians about statistics.
So, and just one other issue I wanted to ask about is, this was not only a multi-center trial, but it was a multinational trial.
That's correct.
So in your analyses, was that accounted for in any way by treating that? I mean, because these are very different healthcare systems.
Yes, that's a great point, and we did not adjust for that, although in terms of the results, it did not make a difference. But we have looked, there are different rates by country, and we had someone who was going to write that paper, and I don't think ever did, but there's definitely differences in the healthcare systems. the patients that were in Ecuador were being seen by the best neurologists in the country. And so he had no problem recruiting because everyone wanted to be seen by him. Whereas in the US, you know, we had different populations. And so it's definitely an issue, and it's definitely a concern, and it's really an issue and concern for many multinational trials that we have very different health care.
And it's mostly ignored.
Yes, it is. It is. You're correct. Neil, you had a question?
When you have these adaptive study designs, do you have to take into account at the point where the adaptation occurs? Because you're disclosing to the staff and you're disclosing to the patients also that we're extending this trial, so they may think something's going on, and I wonder if that could influence behaviors.
That's a good question, and that's part of the hesitancy on the part of government agencies to approve studies that use adaptive designs, because you are telling someone something about the trial. For sample size re-estimation, actually, it's been proven statistically that there is not an impact statistically on where you do the sample size re-estimation. What is impacted is how much information you have at that point along the trial, right? So there's always variability around it, and early on there's a lot more variability. And so if you do a sample size re-estimation early, you don't get as much information from it. But in terms of the impact on potential bias, I don't know that there's a lot of literature about that. I mean, in the cases where you're dropping arms, it's less of a concern because that just tells you those arms were not effective, and so you're going to focus on the ones that maybe were more effective. But maybe there is. It's a good question. There's not literature on that I know of. Yeah.
Is there anything specific you could tell us about what your team did to work with the study fatigue that you mentioned?
Yeah, so Dr. Benavente, Oscar was really good at, he's a very good communicator. And so we did, at the International Stroke Conference, we had meetings where we encouraged the coordinators and the PIs to continue in the study. We sent reports congratulating them. So oftentimes in clinical trials, we use reports sort of punitively and say, look, you're not doing as well as the other sites. But we tried to use them in a more positive way to say, look, we're doing so well, we're getting closer to our target. Just regular communication. We had the best clinical coordinator I've ever met. She was outstanding. She knew all the site coordinators. She talked with them regularly. We had regular trainings with them on the data entry end and on the data systems end to encourage and remind them. But I will say, one of the nice things with SPS3 for being such a large study was it was a really close-knit study, and people knew each other and enjoyed each other's company, and so we used that community to really encourage people to complete the study. Yeah.
Sort of a real-world question, and there's a specific for your study, and then a more general one. So how did you control, because in the US there's multiple forms of treatment, primary treatment for the patient, and if you're looking now internationally, you're going to have even greater variability of the initial management of the patient. So was that taken into consideration at all?
It was not.
Okay.
So I think the average time from stroke to enrollment was 3 months. And so we were taking over management then. So I know there's a lot of discussion about the acute care of someone who had a stroke. So we were intervening at about 3 months.
Okay, because for instance, there's a big stroke center at Drexel that has a special technique they use.
Oh, really?
That's not standard.
Okay.
And then the second part of that would be, how do you control for the fact that, especially in a longer-term study, guidelines may change and diagnostic tests may change, and patients that are in an exclusion criteria may no longer be applicable?
Yeah, we were pretty lucky in terms, because we were looking at such a specific stroke subtype, there's just not a lot of literature in looking for stroke. So we were pretty lucky that the guidelines didn't change much. There were some guidelines that came out in other stroke subtypes saying that whether you should or should not use dual antiplatelet therapy. There was one thing that we saw, and I'm trying to remember, that had to do with like a GI medication that was counterindicated for Plavix during the time of the study. And so we did have to make changes and make sure all the sites knew not to prescribe that or make sure their patients weren't taking it.
Right, but that's for your study.
But we were pretty lucky, actually.
Yeah, but in the general case, that's probably not true.
That's true, especially these long-term studies, and you know, I mean, we had evidence of changes in recurrent stroke rates, but besides that, we were pretty lucky that we didn't see things coming in the literature that caused concern either for us or for our DSMB. I mean, the dual antiplatelet therapy, I think every time we went, that Oscar did have to make the argument this is still relevant.
So it seems that now, as you mentioned, in applications, people are really planning an adaptive clinical trial. So I guess many of the problems that you've seen, regarding changes in forms of people or personnel, should be improved or should not be as bad as what you experience.
Yes and no. So you bring up a good point, but the problem is you can't plan what's going to happen, right? So when we propose an adaptive study, we present a set of simulations of cases that might happen, and this is what we will see if we do it. But then in reality, what happens may not be any of those cases. So we have to hope that we're implementing it in a situation that it still leads to good operating characteristics, good power, and good type 1 error rates. But definitely there is some more attention to the impact on cost because you have to budget. Although if you do a sample size re-estimation and you decide to extend your sample size, then how do you ask for that? You don't know. And NIH doesn't want to give you any more money than they have to. So there is a lot of discussion about how to really implement a good adaptive design a priori, fund them, and plan for them. So that's a really great question, and there's a lot of discussion about that at all levels. I mean, you go to the clinical trials meetings, we talk about that. We talk about it on study section as well. When we're reviewing these grants, we say, have they accounted for this, how these changes might occur, and what that means? So it's difficult. I mean, I guess I'm happy that we're starting to recognize that there are statistical ways, that there are ways that there's statistical evidence to support their use, but I'm, I guess, a little nervous that we're trying to use them too frequently when maybe we're really not gaining much by doing that.
So you mentioned there was these major bleeds, increase of adverse events. Within the discussion about whether to continue the trial, were you able to estimate how many adverse events you would actually see and weigh that against the increase in the power that you have?
That's a good question. So we didn't do that. We did look at how many events, primary events, we would expect to see, but we didn't look at how many additional safety outcomes we might see. That's a really good question and a really good point, actually. You should have been on the DSMB.
Yeah.
Other questions? Well, thank you again for the invitation.